For centuries, the world believed syphilis stormed into Europe with Columbus, an invader unleashed by the New World. Historians clung to the 1495 Naples epidemic as proof. But when a single fragment of ancient DNA surfaced from Lithuanian bones, everything changed. The timeline fractured. Evidence pointed to a disease haunting humans thousands of years before Columbus ever set sail. If the official story was wrong, just how deep do these origins go? And what does the truth mean for everything we thought we knew about medical history? In the winter of 1494, rumors of a terrifying new affliction swept through Naples. Soldiers returning from the French invasion staggered into the city with bodies covered in erupting sores, flesh sloughing from their faces and limbs. Within months, the disease spread with a speed and severity that stunned physicians. Chroniclers described men who had been healthy one week, unrecognizable the next. No one in living memory had witnessed anything like it. The epidemic killed thousands, leaving survivors disfigured and isolated. European doctors, desperate for answers, scoured medical texts for clues. Yet the ancient treatises of Hippocrates and Galen made no mention of such symptoms. The closest parallels, leprosy, plague, did not fit. By 1496, the Venetian physician Niccolò Massa wrote that the disease seemed entirely new to Europe. In 1530, the Italian scholar Girolamo Fracastoro published a poem, Syphilis Siv Morbus Gallicus, giving the disease its infamous name and cementing its reputation as a foreign curse. Fracastoro imagined a shepherd, Syphilis, struck down by Apollo for angering the gods, a story that echoed the public's sense of punishment and fear. The absence of unmistakable syphilis cases in medieval records added weight to the theory that it had arrived suddenly. No reliable reports described the telltale ulcers, bone deformities, or congenital cases in Europe before the Naples outbreak. Skeletal remains from earlier centuries with pitted skulls or shin lesions were usually attributed to leprosy or other chronic illnesses. Even as far back as Pompeii, ambiguous bone changes were dismissed as unrelated. By the 18th century, London's hospitals overflowed with syphilitic patients, but the earliest clear evidence still pointed to the late 1400s. The story fit the anxieties of the era. Columbus's first voyage had just returned from the Americas. The idea that his sailors brought back a devastating new disease spread quickly, reinforced by the suffering on the streets of Naples. The so-called Columbian hypothesis took hold among doctors, clergy, and the public alike. Blame shifted across borders. Italians called it the French disease. The French blamed Naples, and so on, each group eager to see the scourge as a foreign import. For centuries, this narrative went unchallenged. The timeline was neat, the evidence appeared clear, and the story offered a convenient explanation for a plague that seemed to come out of nowhere. Yet beneath the surface, the historical record was full of gaps and uncertainties, with no definitive descriptions of syphilis. Before 1494, scholars had little reason to doubt the accepted story or to imagine that the disease's origins might reach far deeper into the human past. In a basement laboratory at the Max Planck Institute, a set of bones from a Lithuanian churchyard arrived for testing. The remains, radiocarbon dated to the early 1400s, offered a rare chance to look for traces of the syphilis-causing bacterium Treponema pallidum in pre-Columbian Europe. Dr. Kirsten Boos, a paleogeneticist known for her careful approach, led the team. Ancient DNA work is a field haunted by contamination. A single stray molecule from a modern sample can ruin months of work. As Boos's team prepared the samples, a routine check revealed a near disaster, a cross-contaminated reagent batch. The team halted everything, re-extracted the DNA, and shifted to a single-stranded library preparation method, a technical tweak that had only recently become available. This approach, championed by bioinformatician Dr. Martin Sikora, allowed them to recover DNA fragments that older methods would have missed. The difference was immediate. Instead of the faint signal of a few treponemal fragments, the new method boosted the yield by nearly 40%. For the first time, they could reconstruct a near-complete genome of the ancient bacterium from a skeleton dated decades before Columbus's ships reached the Caribbean. The sequence matched Treponema pallidum, the spiral-shaped organism behind both syphilis and its close relatives. The implications were startling. Here was genetic evidence of a syphilis-like infection in Europe before 1492, at a time when the disease was supposedly unknown on the continent. The Lithuanian genome, authenticated by damage patterns unique to ancient DNA, survived centuries of burial and the chaos of laboratory error. The findings threw the neat timeline of the Columbian hypothesis into question. If treponemal disease was present in Eastern Europe before Columbus, the story of syphilis's arrival in the Old World was far more complicated than anyone had assumed. Colleagues across Europe and North America took notice. The technical leap made by Socorro's single-stranded library prep opened new possibilities for pathogen research, not just for syphilis, but for other ancient killers, plague, tuberculosis, and beyond. Suddenly, the search for the origins of syphilis was no longer limited to the archives and old bones. The laboratory became a place where the past could be read directly from the molecules themselves. The Lithuanian discovery did not settle the debate, but it planted the first major doubt. If the disease's genetic fingerprints could be found in a 600-year-old skeleton, how much deeper in time could scientists look? The door was now open for evidence from even earlier eras, waiting in the DNA of bones buried for millennia. A field camp in Oaxaca, 2022. The midday heat pressed down, threatening to erase centuries-old secrets before they could be found. Dr. Lorena Miro, an archaeologist with a background in both genetics and community engagement, stood over a freshly opened burial. She had just minutes to decide how to preserve what might be the oldest traces of a deadly pathogen. In tropical climates, DNA degrades with every degree rise in temperature. Bones become sponges, soaking up humidity, bacteria, and the chemical fingerprints of decay. Standard protocols, plastic bags, coolers, and dry ice were no match for the relentless heat. Miro turned instead to a method borrowed from forensic disaster sites, microclimate control. She packed the remains in layers of sterile gauze, sealed them in vacuum pouches, and buried them again in a shaded pit, where the temperature hovered several degrees cooler than the surface. This impromptu refrigeration slowed the breakdown of genetic material, buying precious time for the team to negotiate with local authorities and indigenous leaders. The stakes went beyond preservation. For years, researchers had debated the ethics of sampling ancient remains, especially from indigenous cemeteries. Miro's team worked under agreements that prioritized respect. Each sample required written consent from community representatives, and destructive analysis was limited to single teeth or fragments. Dental pulp, nestled deep inside molars, offered the best hope. Unlike bone, it is shielded from most contaminants and preserves a molecular record of infections that ravaged a person's body and life. The Oaxaca fieldwork produced only a handful of teeth, but each was treated as irreplaceable. Back in the lab, Miro's samples faced another hurdle. Tropical DNA is often fragmented beyond recognition. Advances in sequencing technology, first pioneered for ancient plague and tuberculosis, now allowed for the recovery of even the most degraded pathogen DNA. Single-stranded library preparation, a technique refined just 3 years earlier, could amplify fragments as short as 30 base pairs, lengths that would have been dismissed as noise a decade ago. The approach required specialized cleanrooms, ultraviolet sterilization, and negative controls at every step. Even so, success was never guaranteed. Most samples yielded nothing but silence. But in one case, a molar from a pre-Columbian burial produced a faint signal, a few molecular echoes from the spiral-shaped bacterium that causes treponemal disease. The result was more than a technical victory. It proved that, with the right blend of field ingenuity and molecular precision, ancient pathogen DNA could be recovered from some of the world's most challenging environments. Miro's tactics, both in the field and in the lab, set a new standard for authenticity and ethical collaboration. They opened the door to discoveries that would soon redraw the map of humanity's oldest diseases. In a rock shelter above the Sabana de Bogota, the remains of a young woman lay undisturbed for more than 5 millennia. When archaeologists uncovered her skeleton, nothing about the burial suggested the presence of a notorious killer. Yet inside a fragment of cortical bone, scientists found the oldest known traces of treponema pallidum, the spiral-shaped bacterium behind syphilis and its relatives. Radiocarbon analysis placed her death between 3600 BCE and 3400 BCE, thousands of years before the first written records of the disease. The genome extracted from these ancient bones, labeled TE13, did not simply nudge the timeline for treponemal disease. It shattered it. Genetic sequencing revealed that TE13 belonged to a branch of treponema that split from the ancestors of modern syphilis, yaws, Bajel, and Pinta nearly 13,700 years ago. This was long before the rise of agriculture, at a time when human communities were still organized as mobile bands of hunter-gatherers. The bacterium's genetic family tree, reconstructed base by base, stretched backward into a world without cities, without borders, and without the dense populations that would later drive epidemics. The implications reached far beyond Colombia. The presence of treponemal DNA in a 5,500-year-old skeleton anchors the disease deep in the prehistory of the Americas. It also casts new light on the movement of people and pathogens across continents. During the late Stone Age and early Bronze Age, trade routes crisscrossed the Americas, linking distant groups through the exchange of obsidian, shells, and information. By 3,200 to 2,800 BCE, these networks were already in place, allowing not only goods, but also microbes to travel vast distances. Disease did not wait for the arrival of ships from Europe. It moved with every migration, every gathering, every shared tool or embrace. To confirm the antiquity of the TE13 genome, researchers relied on a combination of radiocarbon calibration and Bayesian divergence dating, a statistical approach that compares the genetic differences between ancient and modern strains to estimate when they last shared a common ancestor. The results were unambiguous. Treponema was not a recent invader, but a companion to our species for thousands of years. The neat timeline that once placed syphilis's arrival in Europe at the dawn of the Age of Exploration now stood in ruins. Instead, the evidence pointed to a far older, more complex relationship between humans and this elusive pathogen, one that spanned continents and epochs, and that continues to shape our history in ways still being uncovered. In 2025, a team led by Dr. Ada Auner published the full genome of the ancient treponema strain known as TE13. This was not just a technical milestone. It revealed a lineage of the bacterium that split from the ancestors of modern syphilis, yaws, Bajel, and Pinta nearly 13,700 years ago. TE13's DNA, painstakingly reconstructed from a 5,500-year-old skeleton in Colombia, contained a suite of genes that allowed it to infect and persist in human hosts. Among these were the TPR gene family, molecular keys that let the bacterium evade immune detection and adapt to new environments. The TPRC gene, in particular, showed subtle differences from its modern counterparts, hinting at a long evolutionary dialogue between pathogen and host. Dr. Auner's analysis suggested that ancient treponema strains may have been able to infect people through skin contact, much like yaws today, rather than through sexual transmission. The presence of virulence factors in TE13 points to a capacity for chronic, recurring infection, a relationship that may have shaped human immunity over thousands of years. Yet the exact interplay remains a mystery. Without ancient human DNA from the same site, there is no way to know whether early populations carried genetic defenses against the disease, or if the pathogen's success lay in its ability to slip past every barrier. The discovery of TE13's genome does more than fill a gap in the record. It exposes the deep roots of a pathogen that has haunted humanity since the Stone Age, adapting in lockstep with its hosts. For clinicians and researchers, the ancient sequence offers a new reference point, a genetic fossil that challenges assumptions about how and when diseases become part of the human story. The discovery of ancient treponemal DNA in a 5,500-year-old Colombian skeleton forces medicine to reconsider its most basic assumptions about disease transmission and evolution. For decades, medical textbooks described syphilis as a sexually transmitted infection, its spread confined to intimate contact. Now, with evidence that ancient strains may have moved through skin-to-skin contact, much like yaws or Bigel, those boundaries blur. The old dichotomy between venereal and non-venereal treponemal diseases no longer holds. Instead, the bacterium's history reveals a spectrum of transmission modes shaped by environment, social behavior, and population density. This genomic record also reframes the timeline for antibiotic resistance. Modern resistance mutations, such as the 23S rRNA A2058G variant, only appear after the introduction of antibiotics in the 20th century. Ancient genomes show no trace of these adaptations, confirming that the arms race between drugs and pathogens is a recent phenomenon. Yet the same sequencing methods that uncovered syphilis's ancient roots are now being used to track the origins of resistance genes in other pathogens, offering a roadmap for anticipating future threats. Ancient DNA has become a universal toolkit for medical detectives. The same approaches have reconstructed the genomes of plague from Black Death victims and tuberculosis from medieval skeletons. Each new sequence extends the reach of medicine, allowing researchers to trace how diseases adapt, migrate, and resurface. The paleogenomic community now stands at the threshold of discoveries that could reshape public health, revealing not just where humanity has been, but where it may be headed. Modern medicine still wrestles with questions ancient DNA is only beginning to answer. As we unlock genetic secrets buried for thousands of years, the story of disease is rewritten, and with it, our understanding of risk, migration, and resilience. The past isn't just history, it's the blueprint for tomorrow's epidemics. The next breakthrough could already be waiting in the bones beneath our feet.